Okay, let's start. Hi, everybody. Nice to see so many people are already on time and online. My name is Jill, and I will be giving this virtual course session together with Dennis, who you also see on camera. We are both postdocs in the lab of Petra Ritter and the brain simulation section here in Berlin. And we're very happy to be entertaining you for the next 3 hours, and hopefully you will learn a lot about multiscale simulations. So this is the third session of the course on personalized brain simulation, and this is a virtual course. So here we have the agenda of the whole course. If you have been with us from the beginning, then you have seen an introduction by Petra, and then last time, which was just before the holidays, we had an introduction on the eBrain's workflows, including a lot of pre-processing and how we come actually from the imaging data until the virtual brain. So there was also some homework for those of you who want to earn credit, and the deadline was, I think, today. But if you still want to do the homework, of course, feel free to do so, and any requests of homework go to Petra. This time we will talk about the multiscale co-simulation. And next time, in 2 weeks, you will learn about the subcellular pathways and brain network simulation. And then it's about stimulation on the 4th of February, where you will see me back again. And in the end, there will be the participant presentations. Details on that will follow from Petra directly. We will have today a short break between 4:30 and 4:40, our time zone, depending on where you are, and which will be after half of the course. And for the stimulation, since I will be back, this would be great if you give us some kind of feedback, if you liked something we used or not, or if you have suggestions on how to improve the course. We are always open to that. So best would be to send an email to Petra, such that we can improve our virtual course on this. So I posted the slides, and I hope you all have access to it. Hey, okay, familiar faces, great. Okay, so overview of today's course. We will start the course now with the first part of a lecture, 45 minutes, and always alternate between lecture, hands-on, again a lecture and a hands-on. So the first part of the lecture will be a general introduction into TVB multiscale. What is it? What can you do with it? And so on. Then we will run TVB multiscale in its very general framework on a first, make your own first simulation. Hopefully that will all work out. You should gain access to everything, and we will see that in the hands-on session. Then a 10-minute break, and after the 10-minute break, we will have another lecture part where I will present to you together with Dennis 2 specific use cases where we have already used TVB multiscale and where we are actually performing ongoing research on. So this will be really fresh results and really nice, I think, connection between education and research. And then in the second hands-on part, you will be ready to actually run one of these use cases yourself and hopefully also experiment a bit with it. And then for the homework, you will have more time and also the full resources of the eBrain's collab to then run even more simulations with this nice use case. Okay, so we wanted to make this session a bit interactive, or as much interaction as it's possible virtually. So we have a first question for you on menti.com. So I will post this as well, our Menti questions, again in the chat in case people joined later. So we would appreciate if you go to menti.com and you fill in this code and you tell us what is your background and what did you study. Because this can be very interesting for us, for the hands-on session as well, to know a bit of your background, and to know a bit our audience, to whom are we actually talking here. And I will post in a few minutes results of this in the chat. And we have, this is not the only question, we have 4 questions. So there's another one on what was your experience with, do you have any experience with TVB simulations? This will also be very interesting for us, knowing it before the hands-on session. So we know we've given you a bit of suggestions of how to prepare today's session, but don't worry if this hasn't worked out for you. Then it's all anonymous. You can just tell us what's actually, your experience with TVB. And the same goes for Nest. So that's the other software you will learn more details about. Dennis will explain them to you in a minute. And we would like to know if you have any experience running Nest simulations. So I see already people responding, so it seems to work. That's great. Ah, awesome. I'm happy it works. It's all a bit experimental for us today, trying out these interactive virtual things to know more about you as well and to let you participate as well. We will see if that all works out or not. And the last question would be, I will present to you some use cases and Dennis as well, and tell you a bit where we can apply TVB multiscale. But during the whole session, this question will be open and will hopefully create a beautiful word cloud in the end. So you can also wait to answer this question, or you can answer it multiple times later. Just give us some keywords on where you think this software framework, TVB multiscale, can be applied and how this can actually be put into action. Okay, I will hand back to Dennis for an introduction and making a link with the last session.
Okay, hello everybody from my part as well. Let me, you gave me back the keyboard, right? Okay.
Yeah, I don't know how to do that, but I did it somehow.
Okay, right, so hello everybody, and I'm gonna do some of the nasty work today because it's a lot of material for today's lecture, so bear with me a little bit and feel free to ask questions in the chat and Jil will convey them to me. So we're gonna start with remembering some of the things you must have heard already in the first two lectures. Because before we discuss what is multiscale brain modeling, what's the use of it, how we can do it, we should remember a little bit what is brain modeling in general, what is modeling with TVB, what is its purpose and its ways of working, and in the end of the day to see the scale where TVB stands within this multiscale effort modeling. So to start with the goal of brain modeling, we can see it as a bridge linking between neuroimaging data or other kinds of data we collect from the brain and theory about how the brain works, the processes and mechanisms of brain. During the last years in neuroscience, we collect tons of data. They can be structural data from MRI, there can be functional data like fMRI, EEG, and other neuroimaging data. And it's very difficult to be able to summarize this information and make something out of it, understand better, create theory about the healthy brain as well as the diseased brain that would lead to therapies. And so brain model stands between these two, and it's supposed to be a tool that can help us do this. Now, one way that this can work is through simulation. Right? Let me see why I can. Okay. So I'm not saying that brain modeling can be used only through simulation, but this is a very proliferate way. The idea is that we put as much knowledge we already have, as well as some of our theoretical assumptions in the mathematical model of the brain, as well as in the structure of it. We simulate the brain, we generate neural activity. From there, we can also generate neuroimaging data activity, virtual neuroimaging data activity, and eventually compare it, some kind of statistical comparison with empirical neural activity, and either confirm or discard our assumptions and hypotheses, scientific assumptions, or eventually modify them, since we can always go back and change our model, either its structure or some parameters of it, optimize it, and get closer to describing the actual processes and mechanisms of the brain functioning. Now, in particular, when it comes to TVB, this takes a shape, the following shape. On the one hand, we have a lot of structural data. The data that we're mostly interested in, as you will see also in the following, is data of large-scale connectivity. In other words, the white matter tracts that connect different regions of the brain. However, so we are more interested in the strength of these connections that gives rise to a weight matrix, as well as the length of these connections, because given a transmission speed, they inform us about the time delays of communication between one node of the region and another, or one node of the network and another. And these time delays are very important for the global dynamics of the brain. However, we also collect information about volumes and surfaces of the brain through MRI measurements, such that we can reliably generate from the simulated neural activity, neuroimaging activity like fMRI, EEG, etc. This virtual neuroimaging activity is that, as we said, compared to empirical data and help us refine our model. So our model stands somewhere between the structural data we generate at the beginning, and it's a precondition of this kind of modeling, and the neuroimaging data. Mind also that this kind of data and this kind of modeling also opens the door to personalized modeling, since this data can be collected from different subjects, patients, etc., and therefore we can make personalized models, and this has a lot of applications in personalized medicine, etc. Now, okay, so however, today, as we said, we're going to talk about multiscale modeling. What do we mean by this? Well, we can consider at least 3 different scales of the very complex system called human brain, and these 3 scales would be the scale of the whole brain, or what we call a large-scale brain network, a network connecting nodes all around the brain. And of course, we can have different variations about the parcellation of the brain, how big these regions would be. Sometimes we work with like 100 nodes for the whole brain. Some other times in TVB, we can make simulations on the surface of the brain where we can distribute thousands of such nodes. But in any case, each of these nodes represent a network of neurons of large numbers, like thousands or millions or more. Now, at every node of this network, we assume that we have a so-called mesoscopic description of the dynamics of the brain, of the function of the brain. This mesoscopic description is a description of different populations of neurons and their interactions. For instance, here we blow up one of these nodes, and we can see 2 populations, an excitatory and inhibitory populations, and their mutual connections. So this mesoscopic description of neural population is connected to the macroscopic description, since each one of these regions is connected to all others. Finally, we can think of a third microscopic scale, that one of individual cells. Here, every node of the network is actually a different neuron, which we usually model as a point neuron. In other words, we could even go to a finer scale where we see the soma or the axon, and we have cable equations about the transmission of signals. We don't do that here. So as microscopic scale, we would consider the one of individual point neurons. So here, for instance, the 2 populations, the excitatory and inhibitory, are intermingled. The one is symbolized with black squares, the other with red circles. So out of these 3 scales, it must be clear by now that TVB lives at the first 2, between the large scale of the whole brain network and the mesoscopic scale of its node of the network, its region node of the network. And the way to model this is the following. We assume that the dynamics of its isolated node here can be described by a mean field model, as it's called. In other words, a model of the dynamics of the, let's say, the summarized or average dynamics of the populations of neurons and their interactions. And you must have seen in the last course the basic equation of TVB brain network model. It is a differential equation of this general structure, where it starts with a differential operator that maps a system of equations into its derivatives. So this is a differential equation that will tell us how the dynamical system will evolve in time, assuming an initial condition. Then we have the so-called neural mass model, or what I call the mean field model, a system of equations that describes locally the dynamics of its node and region. Again, and it consists of state variables, so dynamical variables that describe the system and whose evolution in time actually describes the evolution of the dynamics of this model. Now,
Dennis, very shortly.
Yes?
Can you answer the question for everybody that's just come into the chat, why we consider point neurons and why can't we consider real neurons like soma, axon, or dendrite neurons?
Of course we can, and this would be an even finer scale of modeling. We're not doing it in the context of our current simulations with TVB multiscale, and it's a constraint also we get from using, as you see in a while, the nest spiking simulator, which is a simulator that models a neuron at this level, so description of point cells. However, nothing stands between us and really using another spiking simulator with more detailed neural models. But yeah, this would be adding practically probably one more scale, an even finer one. Yeah, so there is no reason not to do it. It depends on the problem at hand, right? And the tools we're using. We need the proper tools to do this. So, okay, getting back here, it's very important to understand that the dynamical equations, the differential equations that describe the local dynamics of so-called mean field equation, the one that dominates the dynamics of a region node of TVB, is described by state variables, and that these state variables usually correspond to abstract or average or summarized description of properties of a spiking network. It could be the average spiking rate of the network. It could be the average membrane potential or currents. In any way, these state variables are summarized descriptions of the activity of thousands or millions of neurons that reside at the brain region node of TVB. Okay, then we proceed to the term of the large-scale connectivity of the whole brain network. We can have some constant scaling all connections, like G here, or later we're going to use the letter A also. We have a connectivity matrix, the one we get, the one we get from MRI imaging. It can correspond to a count, for instance, of the white matter tracks that connect to regions of the brain. Similarly, as we said, the length of these lines can give us a description, a hint about the time delays that need for the communication of the signals from one brain region to the other. Then we have a term for local connectivity, which we will also ignore for today's lecture. That is connectivity around the region, especially it's needed when we do surface simulations. Then we can have any kind of injected input, as when we do stimulation. And finally, until here, everything describes a deterministic dynamical system. However, we can always consider also some stochastic contributions, some noise. that will play in several cases a very important role. So this is the very general model equation of the TVB brain model. We're going to work with one or two models today, and it's nice if you make the mental exercise of linking the questions you're going to see with the terms here. I will also help when I'm describing them. So now we have a happy break for Zil.
Yeah. So we prepared something else for you to get some interaction in this course and see how well you understood the content. We have a Kahoot quiz on the site. So if you go to kahoot.it and you enter this game pin that's displayed here, let me also post it in the chat so you can just copy paste it. And it's just for fun, not for credit, so to use a nickname that we cannot recognize, but in the end, at 6 p.m., this quiz will close and we will hopefully have a winner there. You can always only select one answer, but multiple answers are correct sometimes, so bear that in mind. And the most important thing about this quiz, it's 15 questions in total, and you have as much time as you want for every question, so it's open until 6. And the questions will hopefully answer themselves one after the other in the order of the lecture. So whenever you feel like, oh, this question I don't know the answer to, then just come back to the lecture, listen to us, and see whether after a few minutes you will find an answer for it. So at 6 p.m. our time, so which in 2.5 hours, this will close, and then you hopefully should be able to answer all the questions. And yeah, we will have a winner.
Okay, thank you, Zil. Okay, so we saw the scale where TVB resides, and I hope that in this third lecture we had already persuaded you that TVB is quite unique as a simulator, exactly because it models the brain at a scale that is relevant to neuroimaging data. In other words, we need the modeling of the brain at the large scale if we want to explain EEG, fMRI, and link it to structural MRI data. However, on the other hand, we know very well that there are other cases where we need a more detailed modeling at the fine scale, of spiking networks and spiking neurons, as when we study specific mechanisms where, for instance, where we need to consider also short time dependent plasticity, etc. Now, in most cases, when we model spiking networks, either we abstract completely on the rest of the brain, or we consider a random input like noise or an oscillatory input that supposedly represents the rest of the brain. Now, this is not always enough. You will see in the second lecture some use cases where we really want to place those spiking networks in a more realistic spatiotemporal context. We want them to receive inputs from the brain that have either a temporal content, like in terms of oscillations, or a spatial content in terms of from which region comes which input, or both of them. And in this case, we really need to place such spiking networks in the context of a whole brain network. This is one general case where such multiscale modeling is necessary. Now, how can we do this? And this is a very important slide, because the whole lecture today will go around these concepts. And although I'm going to tell you a lot of complicated things, I will insist, and the homework also will insist on that, will depend on that, on the concepts we're going to see at this slide. So how can we bridge the scales? On the one hand, we have a network of brain region nodes, and we have seen that at each node, the dynamics is described by a mean field model by state variables that each one of them summarizes in some way some activity of a population of neurons. On the other hand, we have spiking networks that are also grouped in different populations that interact, and which we can also, first of all, assign them to specific brain region nodes. This is necessary. We need to know that spiking network A is at one region node or some other is another, so that they have a relationship of connectivity, large-scale connectivity with a specific weight and time delay, as we have seen for TVB. So first of all, they need to have an assignment to a brain region node. The second thing is that we already know that we can have statistical descriptions of the activity of a spiking network. For instance, we can compute the average firing rate or the average current that will flow out of it or towards it, etc. This means that we can bridge the two distance scales through exactly this summary descriptions of the activity of populations of neurons. So on the one hand, we're going to have the state variables that represent populations of neurons and their activity. On the other hand, we can have statistical descriptions of the activity of population of spiking neurons. And there is where we can bring them together. Now, in order to do this, we're going to open a parenthesis here. We have been working on a software, on a TVB software. It's part of the TVB ecosystem. It's called TVB Multiscale. And you can find it at GitHub. I'm going to show you shortly. There are also Docker images which you can download and run. And I'll show you a little bit here and then go to the GitHub website for more details. So the general structure of it consists of a package called TVB Multiscale Core, where the most important parts are the following two. This is in red. Whatever is red corresponds to descriptions of the spiking networks. Whatever is green corresponds to descriptions of the TVB. And whatever is black either relates to the interfaces between the two or it's other tools that we don't need to pay too much attention at this point. So at this core package, we have, first of all, a package called spiking models. This is a set of classes aimed at describing the spiking network at different levels of description. So first of all, at the level of a region node, some devices that can be used for stimulation and measurement, then on the level of the whole network. There are also classes to describe a population, etc. Then in black, there is a package called interfaces, which is exactly the classes we need in order to exchange signals between the TVB and the spiking simulator, so between the scales. And in green here, you see a package that the use of it is to help the user build a TVB model that is more specific to making multi-scale simulations. Now, all these classes have also their builders. In other words, classes that you can configure in order to build specific models. So there are default builders that allow the user can change them manually, or also once the user, an expert user builds his own configuration, he can inherit the default builder, program his own builder, and therefore represent a new model. There are also other packages for data analysis, plotting, writing, but this is not so important at this point. Now, all these general classes at this level of the core package have to be reproduced for interfacing with any spiking simulator we want to interface. Currently, we have interfaces to two spiking simulators. The one is Nest, and specifically its Python interface, PyNest. This is a simulator, as we said, it's quite state-of-the-art. It's a simulator for point neurons. And we have recently also added a first version of an interface to Anarchy. This is another also point neuron simulator, but it is one where it is very easy for the user to type his own equations in order to make a neural model. And then it compiles the models and it can run quite fast. So it has some different characteristics than Nest. So each such simulator has to inherit and replicate, by making them more concrete, the classes of the spiking models and the interfaces. So we have nest models and interfaces from TVB to nest and the other way around. And we have anarchy models and respectively interfaces. And also the specific builders that help us build those models and set up the simulation. Finally,
Dennis,
there are, yes?
Oh, sorry, I thought you were finished with the slide. After the slide, maybe you go back to the first slides where we posted the questions from Menti, because people answered. So we have an overview of how I posted it in the slides already. You just go back to the beginning slides and we can talk about that for a sec before you dive into the next slide.
All right. I will also go a little bit to the GitHub page and then I will let you know when I'm done, okay? So finally, there is a package using Elephant software, which is a software that can help us do spike statistics and convert spike trains into rates, which is important for the interfaces you're going to see, and or generate do the other way around, make the conversion from the rates to spike trains. Anyway, let me show you all this also at the GitHub website of the Virtual Brain. Here is the package, TVB Multiscale. And you can see the structure we talked about. Here is the TVB Multiscale package, and inside you can see core, TVB Anarchy, TVB Nest. Let's see now the TVB Nest. Inside you have interfaces. So classes that help us link nest and TVB in different directions. And you have nest models, which is a representation of a population, of a region node, of a network, of a whole brain. So this is like a wrapper on top of PyNest, which can be used by users who are not specialists in nest, right? Users that come from the TVB side mostly. Yeah, Jil, please go on. You want me to go back to the questions? You're muted.
Yes, please. Yes,
okay.
Please. I can also do it, but I hope you can see the questions with the answers now.
You have changed them? Okay.
Yeah, it should. Oh, no, it doesn't work. So
maybe
if you
I need
close,
to refresh.
Yeah,
if I refresh the page.
And maybe close the full view. Yeah.
Okay.
Yes, you can see them now. Okay, just wanted to share that we have a lot of computation neuroscience students here attending today, and everything else is quite equally distributed, except for the social science, they're underrepresented. But it's great. So we have a very diverse group of people today here. So we will keep that in mind for the hands-on session. And at the next part with the TVB experience, we have some people who ran TVB simulations before, so that's great, but not everybody. So don't worry if you are in the little bit or not at all group. And for the nest, it looks a bit worse. So Dennis, next slide. Nest needs a bit more attention.
Right.
But we will get people hopefully motivated to go on with this spiking neural network software. Okay, please continue.
We're gonna see it, right? Okay.
Yeah, we're gonna see the last question later.
Okay, I see that we're running a little bit late, so I will try to speed it up a little bit. So before we go to more details, I want to tell you what is the default workflow for building a multi-scale TVB model and simulating it. So first of all, one has to set some basic configurations. Then one builds the TVB model and simulator. This step would be pretty much identical if the simulation was only TVB. Then one has to build the spiking network model, in the case of today's lecture, just the nest model. Then it is the point where we're going to insist more, and the homework is going to be about it as well. In other words, how to build the bridge between the two models, the spiking model, nest model, and the TVB model. Finally, we simulate, and in the end, we gather results, analyze, plot them. All this can be done with TVB multiscale software packets, but as we said, we're going to insist a lot on this point here. We're going to go many times through the same information. We won't focus so much although I'm going through it, but there is no time today to talk about the nest simulator or make this very specific. So although I'll go through the other steps, really, we're going to insist more on this. That's why, even before we get to hands-on session, I want to present here in a more general way, for the first time, the possible ways we can actually bridge these two scales, TVB and nest. So we have implemented different scenarios. The first one is an exchange of information from TVB to nest, a TVB to nest coupling, via spike rates. So the idea here is, here on green, we have a node of the TVB network. Whatever is in pink, background, or red, it is supposed to be within the nest spiking network. So let's assume a very general idea, no matter the specific mean field model we use at this point in TVB, but let's assume that there is a model where it has a state variable that represents the mean spiking rate of a population in the mean field model. So the main mechanism we use is that we build a kind of what we call TVB proxy node inside the spiking network. What is this? This is a node of the spiking network that mimics the activity of TVB. So if at time t, a node of TVB has a specific rate, then the specific proxy of that TVB node inside the spiking network will also have this rate and will generate a spike train, and it will couple to any population at any node that we have modeled as a spiking network. So let's assume, for instance, that we have a brain of 100 regions, and we select 2 of them to model in more detail and model them as spiking networks inside as nest spiking networks. Then all other 98 nodes will have a proxy node inside there, which will mimic the activity of their parent node in TVB and will generate spikes and send them to these 2 nodes that we selected to model as spiking networks. Now, how this will work? The connectivity of these 98 proxy nodes towards the 2 spiking nodes, which are modeled as spiking networks, will depend on the TVB connector. So what we have here is a nest device generating spikes, a stimulation device that nest provides for. And this one mimics the activity of the TVB node. It has at every time step a specific rate. We multiply it also with the number of assumed neurons that reside in one population to convert the mean rate to a total rate and generate spikes and send them to the 2 targets, the brain regions and all its populations, excitatory, inhibitory, of the spiking network. The weight of this coupling comes from TVB. This is the structural connectivity connection. The N prime is a TVB node, the N is the nest node. So this is a connection between a TVB node towards a nest node, while there might be also a global coupling scaling all weights. And then these spikes travel and go towards the target spiking node with a specific delay. Sorry, this is the delay that also, as we said, is determined by the length of the tracts connecting these 2 regions. And this spike train, all these spikes have to be sent within 1 time step of TVB. Similarly, another connection might go to the inhibitory population of the same node. So I hope this, as a first explanation, is clear. I repeat, the idea here is to create a TVB proxy node as a nest device, stimulation device. This device is mimicking the dynamics of a specific state variable of the TVB mean field model, in this case a rate variable, and generates spikes and sends them to the target spiking nodes inside the nest network, coupled with the weight determined by the TVB connector and the delay also determined by the TVB connector. Another idea now, following the same general context, is to do this with currents. It could be also that another model or the same model of TVB have a state variable that represents an average current, for instance. Again, we would have to use a proxy node, this time a device that can generate currents. Again, we can multiply with the number of assumed neurons per population in order to convert the average current to a total current, and then again send it to the target nodes that are modeled as spiking networks inside Nest, again with a weight determined and a delay by the TVB connector. So these two cases are equivalent in terms of the strategy, only the kind of information we couple changes from spikes to currents. Now there is a third mode, and this is a little bit different. In this case, we take advantage of the fact that we can actually go and change directly a parameter of the neurons inside Nest. For instance, a current parameter. Every neuron in Nest has an external current parameter which can be used in order to inject some current to this neuron. So here the idea is different. Here the proxy node is the same as the node that is modeled inside Nest. The idea is, so for instance, again, we have 100 TVB nodes, and let's assume that 2 of them are modeled as spiking networks. Now we first compute all the coupling that goes, or the TVB coupling that goes to these two nodes inside TVB. So this is the way TVB already computes the global coupling that is directed towards node N, coming from all N prime, all nodes in TVB, in other words. So you see the structural connectivity, the delay, and the global coupling, alpha, A as well. And then it is applied directly, to its corresponding node that is modeled inside Nest. So in this case, all coupling is already pre-computed in TVB, and then it goes directly to the target nodes that we model in Nest. Again, we have to possibly scale it with the number of assumed neurons. So we could do this with potentials also. For instance, if there was a parameter for potential, we could direct such a parameter inside the Nest network. But until now, we have used this only for currents. So here we don't really need a proxy node inside the Nest network. This is a more direct way. It also is simulated faster. So these are the 3 possible ways we can couple TVB to Nest. In most cases also, we want to have the other direction also, the interface working also in the other direction. However, there is a big difference. From the direction from TVB to Nest, we consider it a coupling. So we had to either create proxy nodes in order to mimic the TVB nodes and couple to the Nest nodes, or as we said, we computed the whole coupling already in TVB and directly projected this coupling to the Nest nodes. However, in the other way around, from Nest to TVB, we consider it an update. Why is this the case? Let's go again. We have 100 regions in the TVB connectome, and let's say 2 of them, we model them in Nest. Although we model them in Nest, they keep being represented inside the TVB. It's not like we create a hole, an empty space in the TVB brain. They are still there. So, and that's important if we want to compute later on EEG or fMRI. We cannot have a hole in the brain where we don't have activity, or we cannot convert this activity. So, so if we really want to have these 2 nodes get information from Nest, the process is one of update. We need to compute the Nest activity for this node, somehow average, summarize it, and go and overwrite the specific activity inside TVB. So the way to do this is we have spike detector or spike recorder devices, as they're called now, inside the nest, and they record the spikes of its population of its region we have modeled in nest. They count them, and then from these spike counts we can compute an instantaneous rate. So we divide the number of spikes to the total number of neurons in the population, and we also divide to get a rate, and we also divide it by the time step of measurement. And in that manner, we get an instantaneous rate, and this instantaneous rate goes and overrides a specific state variable inside the TVB of the corresponding node. I'm sorry. So if this is parahippocampal cortex and we model it in nest, it goes and overrides the activity of the state variable of the parahippocampal cortex in TVB. That's why this is a notion of update or overwriting, not one of coupling. As long as we have overwritten the state variables from nest, the rest of the computations of the mean-field model of TVB can carry on, as well as any links it has with other TVB nodes. So this is like an alien node inside TVB that it's not anymore working according to its own equations of the mean field, but at least some state variables of it are overwritten, are updated by activity coming from nest. So this is the way we go from nest to TVB. Now, as I said, we're going to go through slides like this many times, and we're going to see the code of TVB multiscale software that actually implements these interfaces, and your homework will also be about this. So I repeat some things. What is very important until now is that we keep these concepts of region nodes, whole brain region nodes, populations, state variables that describe the dynamics of populations inside a mean-field model, or spiking networks that implement such populations and their summarized activity, some statistical summary like what you see here with a spike count. At this level of description, the whole lecture is going to carry on. And I hope that in the end you manage to conceptualize all this and do your homework also. So we're getting close to the hands-on session. And in order to do it, we're going to work through the eBrains collaboratory. So there are two ways to start the TVB Nest app that will run a Docker image of the current version of TVB Multiscale, and where we can run our notebooks for the exercises today and for your homework. The same app you can start also from the eBrains collaboratory. So either way, we can start this through this hands-on session. What we're going to do, I'm going to guide you through all steps for all of you that can now connect and carry on with them. Otherwise, you can come back to these slides and do them yourself some other time. So we're going to start the app. We're going to log in. I'm already logged in. You will have to log in with your eBrain credentials. We're going to copy the folder of the course to another folder. So I'm going to show you so that whatever you do is saved and you can find it again. And then we're going to go through the notebook. I will not explain everything in detail because we really want to focus on the interfaces. We're going to run the notebook for different cases of these three different kinds of interfaces I showed you. Let me think. Okay, so now I'll show you how we can start all this from the eBrains website. So even if you start from zero, like let's say you go here. So you can directly follow the links you have in the presentation slides, but I'm going to show you the hardest way here. You can go to services and you can scroll down until you get into simulation and specifically the virtual brain. Once you are at service, the virtual brain, then you have to find the link to the TVB multiscale simulation web app. And as soon as you press on it, what will happen is it will try to start the server for you. Now, I was already logged in. Maybe I need to log out to show you how this happens. So I will start, and it asks me to sign in. And immediately you get here. Also, in my case, the server was already running, so you will have to wait for the server to start. Okay, I'm going to risk it now and try to show you the process. If I stop my server, okay, and now I do everything again. Okay, we have to wait now. I hope this works for me. Launch server. So you have both to log in and your server to launch. Okay, it worked for me. I hope it has worked for you also. If not, just follow my steps, take notes, and you can do it at some other time. So here we see two folders. Or you ask
questions in the chat. Sorry, or you ask a question in the chat in case this is not working for you. I will try to help in the chat.
Oh, perfect. Thank you, Zil. So we have two folders here. They are different. The first one is empty. Well, not for me, but it's empty for you. This is a folder whatever you're doing here will be saved, and if you log again some other session, you'll find your work there. Unlike that, whatever you do within the other folder, if you stop your server and you start it again, it will have gone. So that's why I suggest you do the following. You find here the folder of the course, you select it, and you ask it to move to the other folder. Contributed notebooks. So although it is a move process, if you stop your server and you start it again, it will be here again. So we don't care about this, but once we have it inside the contributed notebooks, yep, we know that it will be if we stop, you do it here also in our the server and we started. So now whatever here, you can save it. Now inside this folder, everything you will see 3 notebooks. We're going to start now the reduced Gong-Wang. In the next hands-on session, we're going to look at the use case of a basal ganglia modeling network. And then there is a basal ganglia homework network where you will have to work with your homework. More information on the homework we're going to discuss on the next hands-on session. There is also a homework document where you have to fill in the answers. So now we start the reduced Gong-Wang model notebook, and we're going to go through it in order to show you how one can design and perform a TVB multiscale simulation. Okay, so here is a slide reminding why we're doing all this. This is the link I showed you earlier for the GitHub location of TVB multiscale. And we are starting with a description of the min-field TVB model we're going to use for this example. So at every node of TVB, we're going to place a model following the dynamical equations you see here. It is a model that has an excitatory population and an inhibitory population. It has 2 dynamical state variables. Both go with a letter S. There is an S excitatory and an S inhibitory. These state variables have, yes?
Can you increase the size a bit so that we see it a bit bigger, the equations?
Yes, give me a second. Is it better
great.
now?
Thank you.
Okay.
Yeah, thanks.
Right. So the meaning of these state variables is postsynaptic gating dynamics. It's the proportion of synapse channels open at any given time. Since they are a proportion, they can be between 0 and 1 only. They are bound on this interval. You see that the excitatory population has a more complicated equation that has some non-linearity here because there is a multiplication between S and another state variable R, while the inhibitory population has a simpler form. Now, both equations receive an input of rate. This is another state variable. Actually, this is a variable variable is
Dennis, could you repeat the last part? I think your internet was down.
Sense of describing the dynamical system. The system has only two state variables, the S of excitatory and this is you can hear me because the connect hope the connection is not good.
Yeah, now we can hear you.
I cannot do anything about this, unfortunately. Can you hear me? I continue.
Yes, we can hear you.
So, right, so these R variables are the variables of the postsynaptic firing rate, and they are given as a sigmoidal function of the input currents. So both for the excitatory and the inhibitor, we can compute them out of the currents that a region receives. So it's the same equation with different parameters for the excitatory and the inhibitory population. Now, those currents themselves are computed through these two equations here. There is so for instance, considering the excitatory presynaptic current, there is a constant input. There is the self-coupling from the same excitatory population itself. And what we see is that in order to convert synaptic gating variables to a current, we need to compute them, to combine them, to multiply them with a current constant. So the synaptic gating, which is a number between 0 and 1, times this current constant generates a current. Then there is the input from the inhibitory population of the same region. Again, there is a current constant here, which is like the feedback from the inhibitory population of that region to the excitatory population. And then there is the global coupling that comes from other brain regions. Again, we need to multiply the coupling with the current constant to make it a current. There is the alpha parameter of the coupling function of Tvb. And this model also has an additional scaling parameter that is applied inside the equations of the model. So we have two, as functionally it's a bit complicated, we have two scalings here that do the same job. This is multiplied outside the model. It's something Tvb does for all connections. And this is multiplied inside the model after this, after the summation of all couplings coming from different regions. And these couplings, as we said, we have the specific connectivity weight between each pair of brain regions and the specific delay determined by the Tvb connector. So you can see some of the elements of the initial Tvb equations we saw. From here to there, we would say that these are the specific neural field, mean field equations of the local dynamics, and then this is the global coupling term. It's quite similar for the inhibitory population. There is the feedback coming from the excitatory population locally. There is the self-feedback. Here, the current constant is just 1, so it's omitted. And then there might or might not be coupling coming from other regions, from the excitatory populations of other regions. This depends on this lambda parameter. When it is 0, we have no long-range coupling coming to the inhibitory population. If it is more than 0, then we have also what is called feed-forward inhibition. In other words, signals from the excitatory populations of one region, M prime, come and couple to the inhibitory population of the region N prime. Everything else is the same as for the excitatory population. So coupling is mediated by these synaptic gating variables. It's computed, multiplied by this current constant to become a current. Then local currents are computed. From local currents, we get to local rates, and local rates enter finally in the dynamical equations of the mean field model. This is the reduced Wang-Wang model, as it's called. And I will not get into details about the parameters. So at each node of the TVB brain, we place such a system of equations. What happens now on the side of nest, of the spiking network? On the spiking network, as we said, each node is a spiking neuron. For the purposes of this simulation, we have chosen a very simple integrated fire neuron with alpha synapses, which is a very frequently used model of nest. As with all, I guess I saw that many of you are computational scientists, so you must have seen many times equations like this of an integrated fire neuron. The main state variable is the membrane potential multiplied by capacitance of the membrane. And there are several currents that enter the neuron. There is the leakage current, there are excitatory currents and inhibitory currents. There are currents that are external. I remind you that I told you that every neuron in nest has a parameter like this, which we can change in order to inject currents. And finally, we have here a term that usually it's not there. I have placed it here to remind you of the way TVB proxy nodes can send currents to a nest neuron. Here, this current is supposed to be generated by a device, a stimulation device, and to mimic the activity of a TVB proxy node. This is why also it undergoes all this coupling, TVB-like coupling with the connectome, the global scaling, etc. Now, analyzing each one of these currents, the leakage current is given by the leakage conductance times the difference between the current value of the membrane potential and the leakage potential. Similarly, for the excitatory currents, we have a conductance, a reversal potential, and for the inhibitory, just the same. So these are the three synapses, in other words. Sorry, the two synapses and the leakage current. And so this is how we get these three currents. The rest remained unchanged. There are actually four more state variables that implement the alpha synapses for the excitatory and for the inhibitory synapses of each neuron. Spikes enter through this equation, and then there is one more for the recovery. I don't want to go into the details. This is not the point of today's course. Anyway, this is the model we're going to use for each one of our neurons. Now, the way it works is that currents come, raise the membrane potential. When it reaches a threshold, it emits a spike, and then the membrane potential is clamped to a value, the reset potential, for a specific time, 1 or 2 milliseconds, for instance. That is the refractory time. Okay, now given these two equations, how can we actually couple the two? So, as I said, I'm going to go many times through the same information. So, in one case, as we said, we have TVB regions and TVB proxy nodes that will take a rate state variable. We have one here, as we saw. We have these variables here that represent the rates for every region, multiplied with the assumed number of neurons, and through a spike generator nest device, send spikes, a train of spikes, to every target in nest, weighted by the TVB connectome and with the delays depending on the TVB connectome. Alternatively, we can use currents, outgoing currents. How can we get outgoing currents? By taking the state variable S and multiplying it by the constant of the current So as currents, we can use nest DC currents and inject currents to the target nodes in nest. So its current is taken like this: synaptic gating variable times the current constant times the number of assumed number of neurons in the population. And finally, the third way is we use the TVB to already compute the total coupling that goes to its nest region. We multiply it with the number of neurons, with current, and we send it directly to, to this parameter for external currents inside the neural equations. So, in other words, the target to send from TVB to nest signals can be either this parameter, or we can set currents through devices that send currents, or we can send spikes through devices that send spikes. We're going to see more about it. And for the other way around, from nest to TVB, we need to count the spikes for every population. And this is how we count them. We divide by the total neurons of a population to get a rate. We divide also by the time step. We create an instantaneous rate input, which goes and overrides an input like this inside TVB. So this is a little bit of a tricky point. What I showed you here are the equations of a TVB mean field node. Especially for the nodes that are represented in nest, we have an additional equation like this, which receives input from nest in this variable, which is computed, as we said, as instantaneous rate. It goes through an integration in order to smooth it out a little bit. And then the resulting variable goes and overrides this variable here, which means that, again, we have 100 regions and the 98 are modeled in TVB that will follow these equations. As you look at them here, the two nodes, though, that are modeled in nest will have a computation of rate not through this sigmoidal of the input currents, but overwritten by this quantity. The rest of the equations will run as usual. So these equations will run for all 100 regions. Just for the two regions of nest, this equation here will be changed, and the rate will be overwritten by the instantaneous rate we compute from nest, smoothed out. Okay, let's see now. I have only 20 minutes. So let's see how we can code for this. So first of all, as we said, there are some configurations at step one. These configurations I will not explain. I have already programmed some of them in this class, but the user can eventually also change them at a later point. So here I'm changing, for instance, just some configurations about plotting figures. So I won't stay on this part. Now, then we really start the fun by, first of all, as we said, creating a model of TVB. This part here would be quite similar, no matter if it is a multi-scale simulation or not. And again, there are two ways to do this. One is to use directly a simulator builder for TVB that I have coded for, and where many choices, many good choices are made. In this case, one would have to uncomment these lines and comment everything below. But I want to show you more about what such a builder does, so I'm doing it here manually. First of all, we have to load the minimal structural information that is necessary for a TVB simulation, and this is the information about connectivity. We don't need volumes and surfaces unless we want to compute some neuroimaging signals that need them, but we at least need the connectivity weights and the connectivity lengths in order to be able to do a TVB simulation. So here, through this command, we can load the default connectivity we have for people to test things on TVB. We can do additional things like some normalization, which I show you above here, the exact processing that is done. So we normalize all the weights by the maximum of outgoing connections. yeah, all this is arbitrary. We can still here do a little bit of more normalization so that we fix the values and the maximum to be 1. And we configure the connectivity so that we can plot it. Then we load a core simulator class. This is not the default simulator class for doing TVB simulations. It is one that is altered by me so that it can work for multi-scale simulations. However, the process would be similar if we did only a simulator, a TVB simulation. We would just import a similar class, and all the rest of the steps would be similar. So here we create an instance of this class of the simulator. We identify the model we're going to use, the equations of which we saw above, and we might also determine some specific parameters of it. These 3 parameters are important. We're going to play with them. It is the global scaling of the coupling that happens inside the model. This is the current constant from the inhibitory population of a region to its excitatory population. And this is the lambda that determines if there is feed-forward inhibition. In other words, if there is connection from the excitatory population of one region node to the inhibitory population of another region node. Here we are setting the connectivity we have just imported and configured above. And here we are choosing the general coupling function. So this coupling function is irrespective of the model, okay? We choose the default one, which is a linear one. That means that after all coupling is computed, summed up, etc., going to a region, it undergoes a function like this. But given that b equals 0 and a has this value, it ends up to be just one more multiplication. This is, sorry, I need to repeat this, what I explained up here, that there are, unfortunately for this model, 2 multiplications that do the same job. This is done outside the model by TVB, and this is done inside the model by the equations of the specific model. So this is exactly what you see here. It is this happens inside the model, this multiplication. This happens outside the model. But we have to remember this because we need to structure in a similar way the connections between the nest spiking nodes. Then we need to set an integrator. In other words, a class that will actually do the integration from one step to the other for the simulation. And the most important things we need to set is the time step of TVB, the time step of integration, and the strength of the noise. Finally, we need to be able to observe the TVB dynamics, and this is done by classes called monitors. There are different monitors for different neuroimaging signals like fMRI, BOLD, or EEG. Here we're choosing only a monitor called RAW. This will record all state variables of the model. It's a very simple one. And we need to specify the sampling interval for the monitor. So after we have done all this, the TVB part of the simulation is all set. And we can actually plot the connectivity, and we get a matrix like this for the weights. And you see we have, okay, let me zoom out a little bit. Right, so this is all connections from all brain regions to all brain regions. And you see we have normalized and sealed all the weights from 0 to 1. And similarly, this is the same for the track lengths. It is in millimeters, so we see that the length here connections are about 25 centimeters, which is normal for the brain. Okay. Now, this is a slide that describes the actual network we're going to build. So we already showed how we build the TVB part of the brain. The large-scale network will have nodes like the ones we saw the equations before. And
can you zoom in again, Batman?
Yes, yes, yes. And
otherwise, it's a bit too small. Sorry.
And out of all the regions, we choose only 2: the right and left parahippocampal cortices, to model in nest. And similarly to the mean field model, we're going to create also for the spiking regions two populations, an excitatory and an inhibitory one. Excitatory and inhibitory, excitatory and inhibitory within each region. They're going to be couplings, self-couplings for the excitatory, couplings from the excitatory to the inhibitory, inhibitory couplings from the inhibitory to the excitatory, self-inhibitory couplings for the inhibitory population. And then there are going to be also couplings between the regions that have to satisfy the TVB connectome in terms of weights and delays. So what we need is first to construct this spiking network, but as we said, always with reference to the region and the population. And then we have to build the connections from TVB to nest, the couplings from TVB to nest, and finally the update of these two neurons, these two regions that, as we said, they always have to update the corresponding nodes they come from in TVB, so that there is no hole in TVB. The integrity of the TVB whole brain network is retained. Okay, so keeping in mind this picture, let's see how we have to do this. First of all, we need to know the indices of the regions we're gonna model in TVB. So we have some code here and we make a list. I can already tell you that the two nodes that we're gonna model here is 33 and 34. These are the two parahippocampal cortices, but here I'm just searching them by name. Okay, then, as I told you, for every model, we have a builder class. Here, since I'm gonna do everything manually, I'm just loading the default builder for the nest network. That's why we call it nest model builder class. The inputs need to be the TVB simulator we just created up there in the first step, the list of nodes that are going to be modeled in nest, the configuration structure, and here I'm telling it to not set the defaults because we're going to do it manually. Then we are setting a very basic parameter here. It's called population order. It is the scalable population size. The idea is that if we want to scale the network in terms of number of neurons, we can always multiply this population order for every different population. And if we want to scale all populations together, we can just increase this number here. Then I'm defining another parameter here that is going to be useful. This is the so-called global coupling scaling. This is the weight of the connections, the scaling of the connections between two spiking region nodes, not within, but between. So this scaling scales this dust lines here between the two regions we model in nest. And we already know from the equations that in order to do this, we need to consider the scaling from the TVB coupling function and the scaling of the TVB model itself. So we multiply the two of them here and we keep them in this parameter. Finally, we're going to also need this lambda parameter that decides if there is a coupling from the excitatory population of one region to the inhibitory population of the other region. The model use okay, we continue by defining for the spiking neurons in nest. And this, as we're choosing the size of population, we want both of them to have the same size, so we just put a scale of 1 here. So the final sizes, number of neurons in each population will be the same for both populations, excitatory and inhibitory, and we get it by multiplying the population order with the scale. Now, the first step, as we said, is to create this population of neurons. Since you're not expert users of nest, you don't want to run nest code, that's why we go through this process of defining this, of configuring this builder to do the job for us. We are building two populations here. We define it as a list of Python dictionaries, one for the excitatory population, one for the inhibitory population. What we need is to assign a label for the population, E for excitatory, I for the inhibitory, the name of the model we want to use, this one, for every neuron. Possible parameters we might want to set for this model. Here we're not changing any of the default parameters. And we also need to determine the scale in case we wanted to change the sizes of one of the two populations. Finally, there is this input. This is used when we want to place a different population at every different region node. You're going to see such an example in the next hands-on session. Here, we want both our region nodes to be the same. So by saying known, these populations will be created in both of these regions. So this is how we configure what populations we want. Notice the important thing here is that we give them a label and we decide and we decide where they reside. So it's very important to build a spiking network with terms of populations and regions. Now, we need to determine the connections within its region. In other words, these connections within the one region and the connections within the other region. Again, we define a list of different connections, each one of which is defined as a dictionary of properties, just like before. We need to say the label of the source population and the label of the target population. So here are the self-connections of the excitatory population. We need to decide on a synapse model, and we use for all of them the so-called static synapse. Now, this is nest stuff that I will not get into detail. Static synapse means there is no plasticity, so it's just a weight multiplied to every spike. We also need to define a dictionary of properties of how we want these populations to be connected. Throughout today's lecture, we're going to use a rule of all to all, which means all neurons of one population are connected to all neurons of the other. And when this is self-connections, we're going to allow also self-connections. So each neuron can connect to itself also. All the rest of the parameters we don't care, because since we chose a rule all to all, we don't care for anything more. People who are more expert in Nest can understand the other options. I have commented them here, but they can be used through our software. So we have defined the source, the target, the kind of synapse, and we have defined also the connectivity pattern. Now we need the weight and delay and the receptor type. The weight is just a weight, as we said, multiplied to every spike, and for the self-excitatory connections, as well as for the connection from excitatory to inhibitory population, we just use 1. For connections from inhibitory population to excitatory, we take this parameter from the Tvb mean field model, which is exactly that. And we put the negative sign because it's an inhibitory connection. And similarly, for connections from inhibitory to inhibitory population, we choose -1, which is again the same as in the Tvb mean field model. As for the delays, we just use a default mean delay, which, I have set it to be 0.1 milliseconds. Fine, the last. So the receptor type configuration here is used when a neuron can have different synapses, and each one gets a different EDG number for every different synaptic port. In our case, there is only one synaptic port, and when the weight is positive, it goes to the excitatory synapse, and when the weight is negative, it goes to the inhibitory synapse. Finally, we have a similar here attribute, nodes, which is used when we need a different kind of connectivity for different brain regions. By setting known, these connections will be created to both regions we have in Nest. Okay, I have to move a little bit faster at this point. At the step 3, we need to define the connections between the two spiking nodes. So these green connections here between the two regions of the spiking network. For this, we need to decide how to take into consideration the TVB connectome information. We do this by defining two functions, one for the weights, one for the delays. These functions take as arguments the input, the source region, and the target region. And you can define any function, but here I use a function I have already coded, which will just take the corresponding entry of the TVB connectome and multiply it with the global coupling scaling we have already defined. Similarly, for the delays, it will take the corresponding entry of source and target node from the delays matrix. And yeah, it will just make sure that it's never smaller than the TVB time step. That's an arbitrary choice I made for the simulation. So having these functions now, we can define the connections we want between the two regions. Again, we need to define a source and a target population. The synapse model and the connectivity are the same. Weight and delay, we're using these functions. The receptor type again is 0 because this model has only one synapse, one kind of port, sorry. And again, we have here arguments for source nodes and target nodes in case we wanted to have different connectivity patterns between different nodes. As we say none here, the same connections will be created in both directions. Now, if lambda is bigger than 0, it means we also want the connection between the excitatory population of one region and the inhibitory of another region. And the rest are pretty much the same, only here the weight function has to take into consideration this lambda parameter for scaling. Okay, so we have defined the populations, the connections with, sorry, the populations, the connections within region, the connections between regions. What we can additionally have is a possible, we can generate some spikes, some noisy spiky input for every region of nest. And we do this in a way specific to nest. We need to define a device, a simulation device that is a Poisson generator to give it some parameters for the rate. We give it a label and we tell it to which populations it should target. So here we say target both populations of every region. Again, we want to apply it to every region. So here we say none. We can give a weight and a delay and the receptor type. This is exactly like connecting two regions. So through this device, we're going to send a constant spike train that will simulate the background activity, let's say, that each region and population is receiving. Finally, we need to determine output devices that will help us measure what happens in the spiking network. Here we do two kinds of them, a spike recorder, so that we can have nice plots of the spikes generated by our spiking network, and the so-called multimeter that can measure some of the continuous time variables of this, of our neurons. Again, we need to determine labels for these devices and connect them to the populations we want to measure. I won't go into more details. All this was in order to build the spiking network. We configure the builder and eventually we say build the nest network. And this gives us a nest network class, which we're going to use further down when we build the interface between TVB and nest. So all this happens, yes.
Should we maybe make a break now and just continue in 10 minutes?
I would suggest to not break it here, but go on.
Okay, however you wish.
Yeah, let's say we go for a few more minutes. We do one simulation and then we continue on the next sensation that's going to be faster anyway. Okay? Okay. So at this point, we have created the TVB model, we have created the nest network model. Now we need to bridge them. Okay, so here we go at step 3, where we need to build the interface between them. Similarly, for the interface model, we need to instantiate a builder. The inputs for this builder are the simulator, so the TVB model, the nest network model, the nodes that we have built in nest, their indices. We give a flag to say that the nodes that are modeled in nest are modeled exclusively, which means that we need to take this activity and update the corresponding TVB nodes. And we also give the information of the population sizes. At this point here, there is an attribute to decide which one of the three scenarios of TVB interface I showed you earlier we're going to implement. And here we're choosing the first one, rate. In other words, TVB will couple to nest by generating spike trains by converting a rate by a state variable. And here we give a flag to say that yes, we want nest to update TVB. Further on, we need to define for this builder some basic attributes of scalings we need in order to convert rates to spikes, spikes to rates, currents, etc. Now, from TVB to spike rate, our rate variable in TVB is already in hertz, so we need no scaling at all. For converting spikes to TVB rates, though, we need to divide by the TVB time step, and we also need to multiply by 1000 because the TVB time step is in milliseconds and we need it in hertz. So here we create a scaling coefficient like that. For the current conversions, if you remember, the state variable we're going to use is one of synaptic gating that needs to be multiplied by a current constant to become a current. At the same time, units of current in this TVB model is in nanoampere, while in nest in picoampere, so we also need to scale by 1000. Finally, we get from the TVB model some of the parameters we're going to need, and we also transfer the global coupling scaling. So, in other words, the constant that will multiply all connections between TVB regions and nest regions based on the TVB connector. And we also define functions similarly for the weights and the delays in order to be able to extract the weights and delays from the TVB connector and apply it also to this interface. So I will describe here only the first interface.
Dennis, can you still hear us? Are you still with us, Dennis? Okay.
And they're contained here.
Dennis, the last 2 minutes you were kind of muted and stuck, frozen. Can you maybe repeat the last 2 sentences?
Right. You mean these things are not explained as well as this?
No, that was fine still.
Okay. Right. So anyway, these are just parameters from the TVB model. We're going to be using the lambda and the global coupling scaling within the model G, and then this is the global coupling scaling, that is the multiplication of alpha and G that you see here. Anyway, yes. So yeah, I'm going to explain the first interface and run it and on the next hands-on session. So as we have said at least 2 times until now, this first scenario, what it does is it takes a state variable from the TVB mean field model that has the meaning of a mean rate, and it drives the TVB proxy node, which in this case is a device generating spikes, in order to send them to the nest, to the nodes that are modeled as spiking networks inside nest. So in our case, if we have 100 TVB regions, 98 of them will have such a proxy node in order to project to the two ones, the parahippocampal cortices that we have modeled in NEST. And we need to scale it by the number of neurons to convert the mean rate to a total rate. We need to scale them according to the TVB connectivity, give them a delay according to the TVB connectivity, and set them to the target populations. So how do we define something like this? In a similar fashion like before, we need to configure our builder. And we say here, if this is a rate interface, so run this code. What we need to define is first the model of the NEST device that will implement this proxy node. Some parameters we don't care about at this point for this model. We need to give a function that somehow will take into consideration the TVB connectome, and another function that somehow will take into consideration the delays. The receptor port, again, it's always 0 for this model. And the most important part here is we have to identify the connection between the TVB state variable and the target NEST population. So the state variable we're using is the rate of the excitatory population, and we will send spikes here, this arrow here, to the excitatory population of its NEST region. And since we want the same kind of interface for all nodes, again, we put none here for source TVB nodes and target NEST nodes. I left last this interface weight. Now, we have several scalings here. These three are taken care by this function here, because the G of the model, the A of the global TVB coupling, and the specific TVB connectome information are all taken care by this function, just as it was earlier when we were connecting the spiking regions among them. However, we still need to scale it with with the assumed number of neurons at its population to convert the mean rate to a total rate. So that's why we also scale before conversion of rate to spikes. We scale it by the number of excitatory populations. Now, if there is a connection towards the inhibitory population as well, a feed-forward inhibition, as it's called, that is, if lambda is bigger than 0, we need to define all this also for the inhibitory population. It goes exactly the same, only here it has a different connection, right? It goes from the excitatory rate to the inhibitory population. And also it has a different scaling because we have to scale by lambda as well. In other words, this lambda here. So this is how we define, and this is the crucial part. I don't want you to remember all details of how we build the NEST network, but it's important that you learn how to convert an interface like this to a code. In your homework, you will have to fill in some of these fields in order to implement the different kinds of interfaces. So I explained here the rate interface, and I will continue with the other two as a reminder also after the break and after Jil. Maybe Jil, I show the other two and then you continue, right? And anyway,
yes, you decide.
Yeah, okay. So give me 2 more minutes to show the results of the simulation. So I will not explain all this because we can do it just after the break. But once we have configured the builder from the interface, again, we just run this command in order to build the so-called TVB NEST model. So that's the model of the interface between TVB and NEST. So we have built the simulator, TVB simulator. We have built the spiking network, and we have built the model that connects them. So now we are ready to configure the simulator, define how long we want to simulate, and just run this command in order to simulate and get the results. And I won't do this now because it's already done for this interface. Finally, there is a lot of code for plotting. This is my code for plotting that uses the TVB multiscale tools, but in principle, you could use your own favorite plotting tools. But anyway, I have here different kinds of plots that I'd like to very quickly explain. So there are plots of the results for the TVB simulation. So for every state variable, the synaptic gating of the excitatory, these are all regions. As you see, they're all between 0 and 1. And this is for the inhibitory population. Then we have the rate variables for the excitatory and the inhibitory. You can already see some of the spikes that are received by Nest for the two regions in purple and brown that they are modeling Nest. This is actually the variable Rin, it means rate input, that actually receives directly the inputs from Nest. And these are the current variables. And these are the same plots, but now in a raster format, where you have on the left axis all regions. So I won't go through them. Now, these are the same plots, but focusing only on the two regions that we have modeled in Nest. So this is the activity of these regions that get input from Nest, which you can see here as spikes, and it is overwritten. And here we end with the plots from TVB activity. And then we have also plots of the Nest activity, the spiking network activity, and we also compute mean rates and correlations. These are the mean rates per region and populations. Don't worry about which is which at this point. Here we have a plot of the spike rasters. So we have the two regions, the left parahippocampal and the right parahippocampal. Dots signify spikes. Each row here is a neuron, and if it spikes, there is a dot, and there is those which is like an instantaneous gray Linus rate computation. So we have the populations of the excitatory population on the one column and of the inhibitory population on the other column. The two regions are in rows. Finally, we have a matrix of the correlations, spike correlations between the populations of each region. Then here we have the plots that we got from the, I need to make this a little bit larger, from the continuous time variables. This is the average membrane potential for each population and for each region, and the activity of the synapses, the ones that receive the spikes, the excitatory spikes and the inhibitory spikes. Then we have the same plots, but this time per neuron. So now on the y-axis, we have each neuron separately, not average values. That's why you see this color-coded plots. I think the first one is the membrane potential, and then the two synapses. So I won't go into more details here. Let's have a break, and we're going to go through the same information for the other interfaces and maybe also a little bit more.
Dennis?
So some of the results a little in detail.
Dennis?
Say that we connect again and then we see Jil the next minutes. Yes?
You were just a bit frozen again. We have.
I can hear you.
Okay, great. Before the break, could you answer the question in the chat about validation of those results? Which is, I think, one that you should answer. It's about what is the validation of the results of the spike correlation of those two regions, or anything about that. I think I can answer as well. You will see in the use cases that we will be presenting to you after the break a bit about the validation, because so far Dennis has shown you the details of the software, and then it will go into comparing this with actually empirical data, which is of course a crucial step for the validation. I hope this is what
I must say validation is a very difficult problem. It will depend on each specific model, right? I mean, there is validation in terms of code. You try to see that your code does what you expect, but in the end of the day, you have to know what you're doing for every specific model, what you expect as a result, and be able to compare the expected result with the actual result. So at this point, we are still working on every use case separately and try to validate the results depending on what we expect from every different model.
Yeah,
so there's no general answer, in other words, here. It depends on every model and use case. For instance, I'm supposed to run a paper, to make a paper exactly on validating these interfaces for a specific model, knowing from theory what to expect.
We have one last question in the chat about this. Thanks, guys, for your questions. Why did the parahippocampal's left neurons actually get spikes? Did you set the pattern of the spikes to this region? So did you tune it, or?
Why do the left parahippocampal cortices receive spikes? I will answer based on this circuit here.
And I think the connectome plays a role as well, of course.
Exactly. So we have modeled these two regions, and they're actually, if you see the plots, all of them are firing. Why would they fire? If you remember, first of all, we implemented a background stimulus that goes to all of the neurons of all the populations. This is what we did here. Where we defined, let me see, yeah, we defined the constant spike stimulus of this rate going to all neurons of all populations of all regions. So they would fire anyway, actually. At the same time, they receive excitatory inputs from all the TVB brain regions they are connected to based on the TVB connector. So all this total input, which is excitatory, makes the neurons fire. And how do we know they all fire? Because when we looked at the plot, give me 1 second here, yeah, they're all firing. This is the left and the right hippocampal cortex, the excitatory and the inhibitory populations. They're all firing. And you can see there is a little bit of delay because delays are different for the left and right hemisphere.
Okay, I think we are ready for the 10-minute break, right? We will reconvene in 10 minutes.
We'll meet again at 5, right? Okay.
Yeah, right. In 10 minutes, if you're in a different time zone, and if you're in ours at 5. You can just stay in the meeting. We will not turn off the meeting or anything. Just turn off the camera, microphone, and be back in 10 minutes. See you guys.
Okay, we're back, I guess. I hope you got a few minutes of rest. I told you I'm going to do the nasty work, and it is quite nasty because it's a lot of material, and we're not showing you TVB simulations or NEST simulations, but actually both of them, so it gets quite complicated. But again, the important concepts I want you really to live with today is really this interface, the question of interface between the two scales and how we can implement it. So I will quickly show you the other few ways to do it. So I'm summarizing that we first created a TVB model, then we created the NEST model, which meant to create populations inside regions. So assign them to populations and regions, that is. Connections between populations but within regions, then connections between populations of different regions. Then we came how to deal with question, and which we dealt at point 3 here. So I showed you we can configure phase that takes rates from TVB and converts it to spikes inside NEST. Let's see now the other two scenarios, as well as the other direction of update from NEST to TVB, which I haven't shown. So if up here we choose current instead of rate, we can just copy and paste here current, then the cell that's going to run is the second one. Again, here we use this concept of proxy nodes, so that every one of the nodes of the regions modeled in TVB will have a little proxy inside NEST, will generate currents and send them to the two nodes, the parahippocampal cortices we are modeling in NEST. So again, we need to define a similar dictionary of properties. We need to decide for the device, the model device that will generate the currents and will play the role of the proxy nodes. Any parameters that go to this device can enter here. Weights and delays are again treated by functions that will take the correct weight from the TVB connectome and the correct delay from the TVB connectome based on the indices of its source TVB node and target NEST node. And that device will also take care of most of these couplings you see here. The one that comes from connectome, the global coupling A that is applied outside the model, and the global coupling that is inherent to this specific model G. However, there is still this coupling, this scaling here. Sorry, the scaling of converting the synaptic gating variable, state variable of TVB to a current is already taken care by this conversion constant we have defined already for the builder up there. So the only scaling left is actually the one that converts the mean current to a total current. However, here we make an additional decision. Unlike potentials that are distributed in parallel and they go to all neurons equally, currents are distributed serially. Therefore, if the total current for the total currents, we need to multiply by the number of neurons in its excitatory population. When distributing it to all neurons, we also have to divide by the number of target neurons. That's why the total interface weight is just 1. Then we have here the crucial dictionary of connections, where we define the state variable and the target excitatory population. And since we need the same interface for every combination of TVB and NEST nodes, we just have known here. If lambda is greater than 0 and we want to link also to inhibitory population, we have to define the same things also for a connection between excitatory and inhibitory populations. So here we change the target population, and also we need to change the scaling now, because we need to multiply by the number of excitatory neurons, but distributed to the number of inhibitory neurons. Plus, we also need this lambda scaling here, but only if lambda is bigger than 0. This is how we implement a current interface using proxy node devices. But a current interface we can also implement by sending directly the total coupling to a parameter of nest. In this case, we just need to copy parameter and make it here, choose it as the way to couple. And here the principle is different. First, the coupling is computed in TVB. It's a coupling based on the synaptic gating variable. It uses the TVB weights and the TVB delays and the global TVB coupling. Then it's converted to a current. The final value is connected to a current. So all the summation coming from different regions happens in TVB, and it's the final thing now that we need to go and change inside every neuron of nest. So this has a different way to configure. First, we say that what we want to send is a current, because we need to multiply with this J current constant. Then we identify the target parameter inside the nest neural model, which is the external current. Again, we need a dictionary of connections from the state variable of synaptic gating of the excitatory population of TVB to the excitatory population of nest. You see here how the link happens at the mesoscopic level of populations. And we want this to be true for all target spiking neurons, regions, sorry. That's why, again, we put an argument none. I left as last one the total interface weight. Let's see here. Again, we need to create a total current out of the mean current, but then distribute it. So that's why this division gives us 1. But now we still have to couple with G, because this G is not computed when TVB computes coupling, because now this is the coupling before it enters the specific mean field model. Therefore, the weight here has to be G. Instead, if lambda is greater than 0, we need to couple also with the inhibitory population of its spiking region. Then, first of all, we need to change the target population here. And then also we need to change the weight. We have to add the lambda scaling, but also distribute the total current to the number of inhibitors. Finally, I didn't say how we get the nest update to TVB. So again, now we need to define an interface from spike network to TVB. And as you remember, we need to count the spikes for every population and every region in nest, compute an instantaneous spike rate, and go and override the respective variable inside the TVB mean field model of the same node. Like this is the same node n, not n prime. It's n, so it's a node modeled in nest, and its corresponding position inside the TVB connector. Now, how we define this? Again, we need to define the spike recorder device in nest that will detect the spikes. Any parameters for the device should go there. We don't want any delays. We want this to be an instantaneous transfer of information. Again, the most important part is the dictionary of connections. Now, what we're saying here is that the activity of the excitatory population should go and override the rate variable of the excitatory population, and the spike activity of the inhibitory population should go and override the input rate state variable of the inhibitory population. We want the same interface for all of our nodes. And finally, the weight here is just 1. All conversions of rates from milliseconds to hertz, etc., are already taken care from the global constant we define here. So we don't have time, unfortunately, to run it here together. But basically, by changing at here one of these three options, you can test the different interfaces, and you will see that the way we have tuned them, you will get approximately the same result for the final, for the activity inside the spiking network. Actually, I will give now, we will turn now to Jil's presentation, but I will run it, and once we get back to the hands-on session, I will show you the result, for instance, using the interface. Sorry, we are here. Yeah, the last interface we saw, the parameter one. So don't worry, because in the next hands-on session, we're going to go through a similar process for another model. So there's going to be a repetition of the same information. So thank you very much, and sorry for the complexity of everything.
Don't be sorry, Dennis. We have another question for your part.
Okay.
How was the Poisson stimulus applied to the brain regions? Is the Poisson stimulus the injected input?
Right. So I'm not sure from the question if it is, there were two Poisson stimuli. One we defined within this, the nest spiking network, just to give a baseline activity to all our neurons. And we did it here by defining set devices that will couple to all of our neurons, irrespectively of what TVB does. And then we also had, so this is a device that, based on some algorithm, just generates spikes on this rate, uncorrelated, and it just sends them to the target neurons. And you can connect it with a specific weight and delay. But in the case where we built an interface between TVB and nest through rates and spikes, we build similar devices. Each one of them plays the role of a proxy node for a TVB node, and similarly for the interval of a TVB time step, and based on the instantaneous rate of the state variable of that TVB node, generate spikes randomly with some algorithm that Nest implements, and sends them to the Nest neurons here with a specific delay and based on the TVB connector. I don't know if all this answers the question, but this is the role of the Nest devices. Nest has this kind of nodes that are just algorithms that generate spikes and sends them to neurons.
Okay. Thank you, Dennis, for all the coding, explaining all the code that you wrote over the last years. The brain is complex, so we need very complex models, and we need to make use of many different softwares in order to tackle all the different scales of the brain. So we are actually kind of out of schedule here a bit, but we will just shorten the lecture part, and and the second hands-on sessions can be shorter as well, because you can also go on and just do it yourself at home and run those notebooks. Okay, so let me take remote access again, Dennis.
I think you have already.
No.
Try to
Can you give it to me again? I'm trying.
All right. So I gave it back to you, and it's yours
now. Great. All right. So just a reminder, on the Kahoot quiz, I can see the ranking. So it is close, and nobody was yet able to answer our questions, which you shouldn't be. So the answers to your questions will come one by one. You can still participate. Just go to kahoot.it and put in this game pin. And in the end, we will reveal the winner. And another reminder on the last question of creating a word cloud, or what applications we can think of for TVB multiscale in the future. Feel free to do this again. We already have a nice word cloud there. But you can re-enter whenever some keywords come to your mind where you can think of applying TVB multiscale. All right. So I will present two use cases of you, which where Dennis and me have been working on over the last months, and which will hopefully give you an overview of how you can actually put this into action and do science with this nice software. So the first use case is about the cerebellum and mice. So the cerebellum is also called little brain, right? It's a very complex structure. It has a lot of fibers in there. Many people who do imaging research often ignore the cerebellum, too complex, just leave it out. Me too, I've also done that. But here we want to focus on it because it contains a lot of neurons and it is important for a lot of brain diseases as well. And why will we look at mice? It's because for mice, we have invasive recording data, which we don't have for humans, right? So we start with mice and later want to translate those results to humans. Okay, so the first use case is about the cerebellum, and it's not only Dennis and me, but we have to give credits to another postdoc and professor in Italy, Professor Egidio D'Angelo, and also our supervisor Petra Ritter. Egidio D'Angelo is from the University of Pavia, and his postdoc Alice Gimignani has been essential, and we actually have done this work together with her. It's work in progress, so you will see very new work where we are still working on. Okay, so why would we do cerebellum and co-simulations? The motivation for this is threefold. So first, we have recently published together with our colleagues in Italy that once you add the cerebellum as a region to a TVB simulation, you get a better fit with empirical data. This was based on fMRI data. So adding the cerebellum for healthy people improves the fit between the virtual brain and the real brain. So that's great. So we need the cerebellum apparently to make our virtual brain closer to the real brain. The second part is that the Italian colleagues of ours from the University of Pavia developed and successfully validated already a nest model, which is on the level of cells, so cellular level model, where every single neuron is modeled with their properties for the cerebellum of a mouse. And they validated this with empirical data. So we have a ready-to-use nest model, very detailed, of the cerebellum. And then, last but not least, we have the virtual brain, as you know, but maybe what you don't know is the virtual mouse brain also exists, where you load data from the mouse Allen Brain Atlas, which is great drug tracing data, giving us a lot of detailed information about mice and their connections in the brain. And the virtual mouse brain, you can just start by starting TVB as well. You can find, if you're interested, there are also a lot of training workspaces on the INCF training space. And yeah, go ahead and do the same, simulating the mouse. So the research question coming from this is that we want to investigate the impact of the cerebellar spiking network on the mean field whole brain dynamics and vice versa. So both directions are interesting and not yet researched. This next would be further in the future to investigate the response to sensory motor tasks. We have this data from mice, and we have some data, fMRI data from humans as well, task-related data. And of course, it would be great to have a model, closed loop even, where we can check whether our virtual brain is responding in the same way as the real brain based on empirical data. And in the future, the cerebellum is important for so many different brain diseases. And a detailed model of it could really help us to target some areas and try out some therapies, clinical trials, and so on virtually on the virtual brain in humans. So that's for the far future. So how is this model actually then built up? We have seen this construct now many times. We have a multiscale model. We have the virtual mouse brain modeling the whole cortex of the mouse. Underlying structure is the Allen brain data, right? So we have all the connections of the mouse brain, very detailed. We model the cortex with the virtual mouse brain. Mean field model, we use for the moment the reduced Wang here and the linear model. And for the cerebellum, we model it with a nest model developed and validated by our colleagues in Italy. So we can just interface their model with our software. And what's their model about? Actually, it's quite detailed and it includes all the cells. You can see it here on the upper right corner that actually all the single cells are modeled in this nest model. Later, you can find the GitHub links as well and try out to run their model. So how is it structured? We have the sensory pathway areas, and then we have a connection in between the cortex and the cerebellum via the mossy fibers. The mossy fiber connections go directly to the deep cerebellar nuclei, which is the output of the cerebellum, but they also go over the granule cells and the Golgi cells, which then go over stellate and basket cells as an inhibitory input to the Purkinje cells. So that's a simplified model of the cerebellum here. And the Purkinje cells, they receive two types of input, one from the inferior olive and one inhibitory input from the stellate cells and basket cells. So there's some integration happening. And a second level of integration is at the deep cerebellar nuclei, which is the output of the cerebellum. For us, then the output of our nest model. And here arrives input from the Purkinje cells and from the mossy fibers directly. This then will lead the pathway to the motor cortex, so back to our TVB model, and hopefully show a response there. Okay, so for the scaffold model, it's validated, as I said, with data. What kind of data did they already use? We had the question about validation, so this can be interesting. They have now modeled this only in nest with Poisson spike trains arriving at the mossy fibers, which are kind of simulating the input of the cortex, and a burst arriving at the inferior olive, which is supposed to simulate the unconditional stimulus of the eye blink reflex motor task. It's a very commonly used experiment for mice, where mice are receiving two kinds of stimuli. One is a small air puff to the eye. This is this unconditioned stimulus. And another one, the conditioned stimulus, is some beep, some audio stimuli. And then the mice close their eyes and this motor response is measured. And these two different stimuli is quite complex to model, and they were able already to succeed in modeling this with their only nest model. So let's interface. First, let's validate their nest model. So we were able to build in their nest model within our TVB nest interface. And you can see here some realistic firing behavior already occurred. Now here from the mossy fibers, going over other cells and ending up here in the Purkinje cells, where the integration of different spiking activities are integrated. Okay, so the scaffold model built was validated in our software. And now the interesting part happens. We interface TVB and nest model. Through the biologically mossy fibers, we have connections here from different areas of the cortex through the mossy fibers to the cerebellum. These different connections come from the underlying connectome, where we use, as I said, the Allen mouse brain atlas. And now we will try out a stimulus at the medulla, and more specifically at the intermediate reticular nucleus in the medulla. And then this stimulus should be causing activity in the cerebellum, and we hope later on, this is work in progress, also activity in the motor cortex. Dennis will tell you a bit about the implementation of this use case.
Right, so you must have become experts by now in this kind of graph. So in this case, we actually used a very simple TVB linear model, but it has a rate variable, just like the one we saw before. And therefore, we used, an interface that converts the rate of TVB to spikes. So I won't say much. We use this scenario of TVB proxy nodes. We computed the total rate out of the mean rate, and then sent spikes with weights, which are not shown here, and delays to the mossy fibers region only of the cerebellum model in nest. And for the update, in order to get some of the activity of the cerebellum back to TVB, we did this only through the DCN region. And yeah, we just counted the spikes using a spike detector, computed an instantaneous rate, and went and overrated only for the regions. This state variable only for this one region of DCN that was modeled in NEST. So it's pretty much the scenario you already showed, but communication only through spikes and rates.
Okay, so what came out of this
I don't know,
interface?
well, practically we get the same result, right? Yeah.
Yeah, we should see now the same result. So
yeah,
just
so
for the
you can.
Okay, you want to start, Dennis? Then I go on with the raster plot.
No, you go on. You first. You can explain the similarity, and then I explain the response. Don't worry.
Okay, we still see the raster plots here as they've shown before, but now we have the deep cerebellum. We have the deep cerebellum nuclei as well here, and it shows some activity. And what's happening at the output of the cerebellum, which is here the DCN, is that it integrates the balance between inhibition and excitation. The inhibition coming from the Purkinje cells here. Purkinje cells, they receive a lot of input from all the previous cells here. And the DCN also receives excitatory input from the mossy fibers directly. So this balance then shows us some spikes in the DCN, which is the output of the cerebellum. So this output is transferred to TVB, and Dennis will explain the TVB side of the results.
Right, so unlike before, that we're just sending spikes artificially generated, this time what we did was to stimulate the medulla in TVB, and then the medulla actually sent more spikes to the mossy fibers here, and this is how all this chain of events was initiated. So you see here the actual time course of the linear stimulus we gave. Oh, come on. And here in blue, it is the response of the DCN. And on the graph below, we see the actual spiky input that NEST communicates to TVB. So actually, we put TVB in the place of the random or artificially made cortex inputs that you show in the previous example.
Yeah, and we see some response already from other TVB regions, but not yet in the motor cortex. So that part is still work in progress. We need to probably increase the strengthen the connectivity between the cerebellum and the cortex, but hopefully more for this in a few weeks from us. Next. I can't seem to click next. Can you?
You have actually. You have gone even
to.
Maybe allow me to take you back. Okay.
Yes, please. Okay, so as a summary for this first use case, we were able to integrate the very detailed NEST model on the cellular level inside our TVB NEST interface, and we were able to validate that it produces actually realistic firing behavior. And the next step is, of course, to include here some nice empirical data as well, because so far we have, okay, realistic firing behavior when we stimulate the medulla, but we haven't compared yet with the right experimental data, which is the next step in the validation. So at the future work, not only validate with empirical data, but also integrate a higher resolution mouse connectome, because the cerebellum so far has only one region, and we want it to be even more detailed, having 33 different regions with different properties. Also, we want to include other regions in a very detailed framework within the virtual mouse brain, for example, the basal ganglia, I will talk about that in a second, or the thalamus, hippocampus, or inferior olive as well. And then the translation of back to humans is equally important to then tackle brain pathologies with this framework and see, when we have a very detailed model of the cerebellum, can this tell us something about brain diseases? Okay, so we have to thank our colleagues here in Italy as well, and Petra for supervision, and the project is called RisingNet. Now, the second use case, let me go through this a bit quicker, but don't worry, you will have a whole session again on stimulation. So this will come back again. Use cases about the structure of the brain called the basal ganglia and Parkinson's disease. So the basal ganglia is a structure deep inside the brain in the subcortical areas, which is often hard to image and hard to really get the fibers there and so on, but we still wanted to give it a try with our software. So this use case is combining this area with symptoms of Parkinson's disease. Why and how is where is the connection here? It's deep brain stimulation. So it has been shown that deep brain stimulation is very successful for Parkinson's disease patients as a therapy. If you don't know what it is, very shortly, an electrode is implanted inside the brain and also a small pacemaker on the height of the shoulder with an invasive surgery. And it's implanted for Parkinson's disease in two candidate regions, either the subthalamic nucleus, or further we would just call it STN, or the internal globus pallidus, the GPI. Both regions have been shown to be successful candidates for improving actually symptoms of Parkinson's disease patients, for example, less tremor in the hands. When you implant this electrode and you stimulate with electrical current these subcortical structures. Okay, so why would we stimulate and how does this look in a 3D framework then in terms of dynamic systems? So we have a diseased brain state here on the lower bottom and we want to bring it to the healthy target state. This is just to give you a vision. So we want to use deep brain stimulation to bring the dynamic regime of the diseased brain closer towards the healthy target brain. And we want to use the virtual brain for that, to try out the different pathways and find out what's the best pathway to actually cure the virtual brain, to then later on try these configurations on patients. So what's actually the challenge here? Why is it not just working for everybody, this therapy? It's that, as you see here, a picture based on the data of 20 different patients and different placements of these electrodes. And you see that they're quite all over the place. They all hit the STN somehow, but not as well. And you can see that back in their clinical improvement. Some patients, the ones where we hit the red fibers here in the picture, showed clinical improvement. And when we hit the blue fibers with our electrical stimulus, we show actually worsening of the clinical characteristics of the Parkinson patients. So this is not what you want. And you see how close blue and red are. So we really need to find the optimal placement for each individual patient, rather beforehand with TVB, in order to then do it correctly, or at least advise the surgeon a bit better and give and offer some advice where to place this for this individual patient. So the research question that evolved from this is, can the model inferred parameters with our virtual deep brain stimulation actually predict this clinical improvement? For that, we need, of course, a very detailed model of the basal ganglia, because a lot is happening in the basal ganglia. We have the electrical stimulus there, we have the first response there, and then everything is transferred to the cortex. So in the cortex, we also not just want to have cortex, but we want to have, for example, a detailed look at the motor cortex, the primary motor cortex, to see whether actually we have an effect there or not. So we could tackle this quite well with our co-simulation framework. So there are so many models for the basal ganglia out there. We needed to choose one. And luckily, we have colleagues in Chemnitz that already validated this model that you see on the slide here, where they use the cortex not as in such detail as we will do in a second, or you will actually do yourself in the next hands-on coming up in a few minutes. But they had the cortex just as 600 excitatory neurons and 150 inhibitory neurons. And they connected it to the different regions of the basal ganglia, the striatum, the STN I told you about, GPE, GPI, thalamus, and then it's projecting back to the striatum. With this model that they implemented, this spiking model, they found quite nice results. And the software that they used was called Anarchy. You have heard this name all the way in the beginning of this lecture. Sorry if sorry, needed to drink something. All the way in the beginning of this lecture, Anarchy is also a spiking neural network software, similar like NEST, and it's short for Artificial Neural Networks Architect. So quite a cool name, developed by our colleagues in Chemnitz. And they showed that they were very close with experiment and simulation fMRI matrices when they used this model and the data of Parkinson patients who had a DBS electrode already inserted. So we started out from this model, but we wanted to replace the cortex by TVB. Okay, so we have a model in TVB that can do this. We can stimulate the STN if we just focus here on the subcortical areas. When we have some nice connectivity there and you see it blinking, this is the graphical user interface of TVB. If you don't know it yet, make some nice visualizations, but we want to link up all the subcortical areas also with the rest of the brain. So how do we do this? Is of course, again, with the TVB Nest interface. And for that, the number one cell in the notebook, as well as the number one thing that you need in order to interface different areas in the brain, is the connectome, right? So you need a weight matrix and a track length matrix. And we got this was published last year by Peterson et al., a very nice atlas containing post-mortem data and detailed DTI data, and also some, as you can see on the pictures, some expert neuroanatomists who were wearing virtual reality glasses and discussing the different fibers in the brain. So it's an atlas that has detailed information on the basal ganglia, and you have access to it as well since we uploaded it on eBrains now. And it's ready to be used in TVB. Okay, so this is in the model. We just replaced the cortex by TVB and the whole connectome. Also, we added the direct connection between the thalamus and the cortex, which is quite important and was not yet considered in the model before. And then the lower part is then modeled with Anarchy. We can model it. We have a TVB Anarchy interface, or with NEST, which we will do in the hands-on session. So we have here again this multi-scale model that you've seen a couple of times now in this lecture, where you have the cortex modeled by the whole virtual brain. Every region has a detailed model, and then you have, okay, please mute yourself if you don't want to ask a question. And then you have the computational spiking model for the basal ganglia, which is modeled in NEST or Anarchy. So a huge model with details on the basal ganglia. And this is just shortly to show you upper and lower picture that NEST and Anarchy, both how we implemented it, we produced the right, very similar and correct spiking results. And as a summary, we integrated a basal ganglia model with a lot of detail on the basal ganglia into the TVB multiscale framework with two different softwares, NEST and Anarchy. And now the cortex, we can have a look inside kind of and see really what's going on in different regions of the brain. And we will now test a first deep brain stimulation on it in the hands-on session. And just a reminder that you can still fill in some keywords if you like to let us know about nice application areas of TVB multiscale. And let's move on to the second hands-on. We have some time, I think, Dennis, until 6, and then it's good to explain homework and yeah, word cloud and Kahoot winner. Yeah.
So we went with quite some detail for this first notebook, where we showed all 3 different ways to make an interface. While Jil was presenting, I just ran the last interface we show, the one termed parameter. I remind you, it's the one where coupling is, sorry, here. recomputed in TVB, and then it goes as a current to modify a specific parameter in the neurons of nest. And the result is a firing pattern pretty much the same as the one we had when the interface was through spikes and rates, which means that we tuned quite well, actually, the interface weights, so that it's an equivalent input from TVB to nest. So you can play yourself with this by just opening this notebook and changing the kind of interface here. And yeah, run it for yourself. When you run rate or current, it takes about a minute because these devices take time to initialize at every time step. Instead, when you use this last interface, it just takes a few seconds. You can see this here. That simulation was just a few seconds, and then it takes some time to plot also. So now we will leave this notebook and we're gonna go to another one. By the way, most of the plots are saved here as outputs, and there are figures. All figures are saved here, and the results are, some of the results are saved also as H5 files that you can read from Python. So now we're going to start this basal ganglia notebook. I will close this. Oh, not I will leave it actually. So now this is the notebook that implements the use case, the last use case Gil just talked about. It's a similar structure. The TVB mean field model we're using is another version, a simplified version of the reduced Wong-Wang model. It's exactly the same equations as before, only this time we have only excitatory populations. That's why we drop the index of E or I. We only have an index for the node, the specific node. It's the nonlinear equation here for the excitatory population, a sigmoidal function to get the rate out of the presynaptic currents. And yeah, I have explained pretty much everything. There is no input now from an inhibitory population because we have only one population. Instead, for the model we used for the spiking neuron, we now use a model we have coded inside Nest. It is a version of the Zikiewicz spiking neuron. It is again an integrate and fire neuron. However, it has a complicated expression here of the membrane potential. It has a recovery variable, a second dynamical variable. And then there are two synapses, one for excitatory connections with Senamba synapse and one for GABAergic inhibitory synapses. There is also some baseline activity, and then this is the external current input. Now, all these three synapses have their own equations here. AMPA receive excitatory. It's just linear synapses. They just AMPA receive the excitatory synapses, GABA the inhibitory ones, and any other stimulation we want to create as a baseline can be directed towards the baseline synapse. This is the equation of the recovery variable. Again, this is an integrated fire neuron. When the membrane potential crosses a threshold, a spike is emitted, and then it is clamped to value C, while the recovery variable is incremented by D, and they stay there for the refractory time. I'm going to show you now only a rate interface, and the homework will be for you to implement the other two kinds, the current interface and the current parameter interface. So for the rate interface, it's similar, just like in the other case. We're using TVB proxy nodes that convert the rate of TVB to spike trains. We have similar couplings here, similar weights, sorry, the alpha weight that is happening outside the model and the G weight that is multiplied within the model. And then we also have the specific connections from the TVB connectome between every different pair of regions from TVB to Nest. Similarly, we have the identical interface to update TVB activity from nest. We count spikes, we compute an instantaneous rate by counting all the spikes divided by the number of neurons per population and divided also by the duration of the TVB time step. This instantaneous rate becomes an input to a linear equation here. It's integrated to get to be smoothed out, and eventually the integrated version goes and overrides, this rate only for those nodes that are modeled in nest. So the actual notebook starts with the configurations as like before. Then we need to build the TVB model. The only difference now is that we are loading a connectivity, a Peterson connectivity only for the left hemisphere by loading 3 different files, one for weights, one for track lengths, and we're also getting the region labels and the center coordinates from another file for every region. And we're also cheating a bit. We are strengthening the connection from thalamus to striatum to see a clear effect in the simulations you're going to see. This is how we create a connectivity, a TVB connectivity. We're also scaling it in a way very specific to this example. I don't want to get into details. Then we are creating the simulator class, and we're just setting the connectivity, setting the linear coupling function with the coupling coefficient we just talked about. We're preparing the integrator with the time step of integration and the lower noise this time. The monitors to observe the TVB activity. And we are also here setting initial conditions that we're loading from a file, such that we get a bigger reproducibility of the simulations. These are all details. I don't want you to worry so much. You can always exercise yourself with TVB simulations. We are plotting here the connectivity is only the left hemisphere. These are the weights. And here at the beginning are the regions that we're going to model in nest. The first 1, 2, 3, 4, 5 regions. I'm giving here an answer also to a quiz. And these are also the trunk lengths in millimeters. Obviously, mouse has a smaller brain than humans. And this is the network we're going to implement. We have 1, 2, 3, 4, 5 regions modeled in nest. The striatum that has 2 inhibitory population. The STN that has excitatory population. GP and GPI also have only inhibitory neurons, and thalamus has only excitatory population. And this is the cortex that we model in TVB that has all the other regions. Now, what you see here is that there is a very heterogeneous network. We don't have the same population at its nodes, and the connections are also different. So that's the big difference now in building this nest network than before. So I cannot go, we don't have time to go into too many details. The steps we're going to follow are, again, building the configuring the populations, configuring connections within regions, then configuring connections between regions, then any possible stimulation measuring devices we want to have in the spiking network, and eventually we're going to focus on how the interface between the 2 models, the TVB and nest, is going to be configured. So these are the 5 regions, the first 5 regions, the indices that we're going to model in nest. We initialize the builder. We set the population order to 200, which is going to be the same for all regions. This time, we're going to do arbitrary scaling for the global connections. We're not going to follow the TVB scaling parameters for this example to work. So we're just sending this one to 1. This means that all connections between regions here, as well as the connections coming from TVB, will just have a scale, a global scale of 1. We choose the specific model, the equation of which I showed you before. We are setting some parameters for all populations from all regions, but then we also need to define some parameters for every different region, the population which are different. We take these values from the respective paper of our colleagues. These are the specific indices for each region. We're going to use them. We are adding up the GPE and GPI indices to have all inhibitory ones, and the STN and thalamus to have all the excitatory ones. The striatum are also different from these two categories. And we need to, now this is the peculiarity of defining a network that has heterogeneity. Again, we have one dictionary for every population. We need the label, the model, the scale, which is the same for all of them. But additionally this time, since we're not building the same population in all regions, here at nodes, we have to define the different regions where each population is going to be built. So by excitatory, we mean both STN and thalamus. So we put here the indices of the excitatory neurons. By inhibitory, we mean the GPE and GPI. So we're putting the indices of GPE and GPI. For striatum, we treat differently. We have a DCN population for striatum and then ISN, which are though in the same regions, in the same region. Finally, the parameters, since they have to be different depending on the node, we define as functions that take an argument the node index, and therein these functions are modifying the parameters for every population. At step 2, we need to connect the populations within region. All the changes we have is the same as in the previous notebook, and there is no much difference here. There are self-connections only for the inhibitory population, as you see. The excitatory population here have no self-connections within the region, I mean. Therefore, we have four kinds, sorry, we're here. We have four kinds of connections. We need to define source and target population. So this is true for the inhibitory GPI and GPE. The synapse model, the connectivity pattern, a weight that we set equal to this value that we defined at the beginning. Yeah, small delays because we are within the same region, and everything goes to receptor 0, all incoming spikes. And this kind of population is built for GPE and GPI nodes. Instead, for the striatum, we have different connections that have to be implemented at the striatum regions. So all inputs here are quite similar with the previous notebook. The difference is that different populations reside and different connections are implemented for different regions. Similarly, for connecting between the regions now, we need to have functions that take as arguments the node indices, and they return the connectivity weight from TVB, and similarly for the delay. And since we have heterogeneity, we have to define all these different connections based on this variety of connections we see with red arrows between the regions here. So I will not go into detail. In all cases, we have a source and a target population, a source and target region, the weight fund, the delay fund, the receptor type is always 0. The synaptic model is just a static synapse, and the connectivity pattern is all-to-all. Finally, we are at this point where we define input stimuli within the spiking network, so it has nothing to do with TVB. In the general case, we're going to send some baseline activity to the striatum, specific to this example, just to, it's an arbitrary choice, just to get the correct dynamics we want to demonstrate in this notebook. So we define a Poisson Generator device, and we're sending a 500 Hz stimulus. We give it a label, and we connect it to the two populations of striatum. Again, no waiting, no delays, and given that this is a baseline stimulus and we want it to go, let me go back to this synapse, this is why we give it a different port. So this is a model with 2 synaptic ports. So this will go to receptor type 1. Finally, we want to be able to measure the activity of the spiking network. So we define a spike recorder and the multimeter, and we loop through the regions and populations in order to create one for each. with a different label. Then we just run this command and we have our spiking network built. But as we said, our focus now is really on the interface, which I have 5 minutes to show you. So it is a rate interface, and we want Nest to update TVB. All these scalings are just like in the previous model. We need to convert synaptic activity to current and change the units. We need to convert spikes per millisecond to spikes per hertz. That's why we multiply by 1000 here. Okay, there must be a small mistake here, but let's ignore it for the moment. So the interface, as I said, is a rate interface. We take the rate of the excitatory population, which is the only one now in the mean field. This one updates at real time for every time step of TVB. A rate generating device that plays the role of a proxy node and eventually sends the spikes with the correct weight and delay to the nodes that receive direct input from TVB. Which are those? Based on this, it has to be thalamus, striatum, and STN. Instead, GP and GPI do not receive direct TVB input. Okay, perfect. So let's see how to do this. Okay, as we said, we fixed the global coupling scaling. So actually what I'm showing you here is not correct. G and A, we don't care because we just scale with one. Instead, we care for the specific weight we get from the TVB connectome, which we get through this function. So please correct me here. G and A do not exist because we just use a coupling of one. So what we're doing now here is, since we have to loop only through the regions that exist, and each one has a different population label, so we loop through the different populations and regions which we need to send input from TVB. It is the striatum and the two excitatory regions, thalamus and STN. And for each one of them, we define a proxy node, which is a device in Nest generating spikes. The weights and delays are given from the TVB connectome. Spikes go to receptor port 0. And the actual connection is always between the variable R. You see that we have dropped the index here. There is no excitatory and inhibitory population. There's only one excitatory population. That's why this is just R. And the target population is given from this looping. It's either the two striatum populations or one of the excitatory, the STN and thalamus. The source node is known because we want all TVB nodes to send such a coupling, depending on the TVB connectome. But the target nodes, it's every time different for every population. When it's for the striatum population, it's a striatum node. When it is for excitatory population, it's either the STN and the thalamus. So it's pretty much the same as before, only now due to this heterogeneity, we need to loop and change the target population and target nodes. And we choose an arbitrary weight, which I have tuned by trial and error here. So we do not derive it theoretically, but we just tune it in practice in order to get our desired result. And actually, your homework is going to be a lot about tuning an interface by trial and error in simulation. For the Nest to TVB update, you must already understand that we just have to create spike detector devices that count the spikes and send it to TVB to override the instantaneous rate variable. So again, we need to loop for populations and regions that are directly coupled to TVB only, not GP or GP and GPI also. We don't care if they are coupled to TVB or not. We just want to observe their activity. So for each one of them, we need to define a spike recorder, no specific weight or delay, and we just need to put the correct linking from the source population label to the variable that they're going to update, which is the variable R input. After we have defined this, we just run this one-liner, and the builder we have configured will build the TVB nest model. A summary of it we can print here. And finally, having our model ready, we can configure the simulator, run the simulation. It will take about 2 minutes. And what we're going to get is actually the resting state simulation. I'm going to directly show you only the spikes that we get. Here we have no DPS stimulus, and that's why you see an activity like this. We have STN, thalamus, GPE, and GPI, and the 2 striatum populations. And you see that there is no much activity in thalamus, and this is a little bit reflected to the striatum. If we see the connectivity here, this is what we expect, that there's some activity in thalamus that goes to the striatum and repeats itself. Now we're going to see the effect of applying a stimulus to the GPI. What we expect is that if GPI fire, what we're going to do is apply an inhibitory stimulus. We're going to make the GPI inhibitory neurons fire less, and then disinhibit thalamus, so thalamus will fire more and send more input to the striatum, so striatum will also fire more. In order to do this, we simulate DPS through as a constant current, DC current stimulus. So I'm uncommenting now this line so that we implement the stimulus simulation. So by commending it, we have the resting state. By uncommenting it, we have the DBS stimulation. This is important for your homework. It is a stimulation of amplitude -200. So we're actually inhibiting GPI, and we give it a label, DBS GPI, and we direct it to the inhibitory population of only the GPI regions. We make it start at time 70 and stop at time 170. So I'm going to run now this, and in 2.5 minutes, we're going to have a result. And in the meantime, I'm going to show you the homework. Okay. So it's running. It's going to take about 2.5 minutes. In the meantime, since we are close to finishing, give me 1 second. So as we said, here we have one more notebook, basal ganglia homework, which I'm going to open. And we also have, sorry, give me a second, a homework document. I don't know if I can open it here. Yes. I'm going to put it aside. Okay, now I have to open it outside. Give me 1 second.
Maybe also go to the homework slide.
Yes, although more information is in the document slide, is not necessary anymore. But yeah, so for your homework, so there is a summary of the homework here, but all this information you can find also in the homework sheet. So let me find it. And ideally, I should now show you the solve homework. Okay, give me a second.
Okay, and if you refresh the presentation then again afterwards, then I can show the word cloud, nice word cloud, and the winner of the Kahoot quiz.
Wow, okay. Okay. So I hope you can see both. Okay, we don't have the result here yet. Let me see. Okay, it's still running, so we can go on for the homework. So the homework is a basal ganglia. On the basal ganglia use case, it's exactly the same notebook we are running now, only now at point 4, where we build the point 3, sorry, where we build the interface. Now we have two that are actually commented here. I will uncomment them. One for a current interface and one for a current parameter interface. And wherever you see a symbol like this, is actual information you have to fill in by using, as example, the rate interface we built for this use case, as well as what you learned from the other notebook on this parameter and current interfaces. So by combining what you learn from the two notebooks, I hope you will be able, to fill in every one of this missing information and run the notebook. So the steps you need to follow are the following. You will fill in first for the current and then for the parameter interface. The notebook is already set to work for a DBS stimulus simulation, so this is uncommented. Most of the inputs here are quite easy to find out by mimicking this interface and also looking at the previous notebook. However, the last one here, the interface weight, is something you need to tune. And what would be the objective of tuning? The objective of tuning will be to get a result with stimulation as close as possible. Okay, or not. So what you see here is that due to the stimulation, there was an inhibition of GPI. So from 70 to 170, this population is not firing, and instead thalamus is firing more. It gives us 6 bars. All neurons are firing pretty much at the same time, so we have 6 bars here. And these 6 bars are also shown as activity in the striatum. So again, what we did was in this network, we applied a negative stimulus here. We made this population fire less, which has an effect. Thalamus fired more, and then striatum also fired more. So what I need you to do in the homework is for the two interfaces, first for this one and then for the other one, first to fill in everything so that you make them work, and then tune the weights, a different weight for the one interface and different weight for the other one, such that in this simulation with stimulus, you get a similar pattern here that will have this pattern of 6 bars firing, and then also reflected in the striatum. After you have made this for each one of them, I want you to go and do a resting state simulation as well. In other words, re-comment the part here with a DBS stimulus and just observe also what is the resting state simulation without any stimulus. Now, although you're going to initially fill on all answers here in order to make the notebook run, by the way, you need to change nothing else, right? You will just modify here to choose a different interface, and you will implement them on these 2 cells, okay? But the crucial part is to tune them correctly to get the correct result. So once you manage this, you come here in the homework sheet and you fill in the replies. So the first one is already given for you. So we have 2 parts, A for the current interface, B for the parameter interface, and it's separated into stimulus simulation and resting state simulation, each part. So the first question is here where you choose the interface, and it's already answered here. There is 0 credits for this answer. All rest of them you have to find at the correct place and fill in the solution. What you're going to send to us eventually is practically the filled-in table here, this document, either as a document or as a PDF. Moreover, there are 2 replies here for which you need to look also in the notebook. In other words, after you manage to tune correctly the interface and get this 6-bar pattern, I want you to copy this picture, make a screenshot or whatever, and come and paste it at the correct place here. So A12 is to copy and paste the picture for the current interface with stimulus. A14 is to do the same for the resting state, and then we have B10 and B12 for the parameter interface with and without stimulus. While also just after the figure, we have here the output of cell 25. So in the homework, this is actually cell 26, I think. Give me 1 second. Right, so this is where you copy paste the figure, and then at cell 27 from the output you have to copy paste the rates of the striatum regions and fill them in here at question 13 for stimulus and question 15 for resting state of part A, and similarly for the part B. Now, all this, I have already filled in an example of how it works for the rate interface that I have already demonstrated. So similar to part A and B, there is part C. And you can see here its different field and how it is filled in and how I have copied it inside the table, and how I copy-paste the pictures with stimulus, with the 6-bar pattern here we're trying to reproduce, and without stimulus, where we have less firing for thalamus, more for GPI, and less for striatum. And similarly, from cell 27, the output, we're getting the mean firing rates of striatum. So it's a bit slow. Yeah. So I just copied these numbers here and I paste them in this solution.
Dennis, I think we have to stop here. We can't give everything away for the homework, right?
It's enough explanation for the homework, but anyway, we have this channel in Slack where you can ask questions. So yeah, I want to summarize that after all this complexity, still what the take-home message is to be able to conceptualize the multiscale model in terms of population activities residing in different brain regions of the TVB connectome. The fact that the scales are coupled through the population activity, either as state variables of TVB or as summarized activity of spiking neurons. And similarly, the interface is built always as a connection between state variables on the one hand and spiking network simulations. And the idea is also of the homework to really be able to configure such an interface, do some trial and error simulations, and be able to tune it such that you get the desired output. And the desired output is also shown here for the rate interface, the 6-bar pattern.
Okay,
I'm going to give you a hint. I need to give a hint to make the life of people easier. Since there is no reasoning behind the interface weights that you need to tune, I'm going to give you a hint. For the current interface, the weight is smaller than 1. It's between 0 and 1. So you have to tune it by trial and error. Every simulation takes about 2.5 minutes, so be smart and know how to dissect this interval. While for the parameter current interface, the weight is in the order of hundreds. It can be 300, 400, 500. Again, you need to tune it, and in order to get the desired outputs. So that's all. We are just 5 minutes late.
Can we go to this presentation again, Dennis?
Yeah, so you fill in the homework, you make a PDF or a document, you send it by email to Petra's Ritter email, okay?